Now let's look at the actin filament animation, actin filament assembly. So if we think that these are the actin proteins denoted with this ball like structures, and this yellow part are the ATP binding region. So actin with ATP binding region are there. So many actin proteins come together and they will attach with each other to make the nucleus. So after the formation of nucleus, they start to add more and more actin towards a particular end, and it is getting higher in a particular end, and it is getting lower in other end. The end it is getting higher is called the positive end, and the end it is getting lower will be called the negative end. Now after few round of this addition and dissociation, it will finally reach and make a polymerized actin filament. But now let's the critical concentration to have the game. Now if we look after zooming the view, so if it reaches the critical concentration, then actin filament will be having a steady state. So if this critical concentration is getting higher, so if the concentration of actin filament is getting high, the critical concentration, then this simple actin proteins will go and elongate. And if it is getting lower than the critical concentration, then some of the actin proteins get dissociated from the actin filament to balance the concentration of the actin proteins outside. That's the basic view. Okay. Now let's come into the regulation of this actin filament assembly. If you can see, this is the actin protein, this is the actin filament. This actin filament production from this actin subunits is tightly controlled via different, or it can be regulated. It can be what we can say affected by presence of different kinds of proteins. So different proteins, some of the proteins will help to polymerize actin, some of the proteins do not help, some of the proteins help to destabilize this actin fiber. Okay, so if you look here in this picture, so let me change the color into blue. Okay, now here we have a protein called formin. If you look in this case, this formin nucleates the assembly and association of the growing actin filaments. So if it attaches with formin, if the formin present in a cell during the assembly of actin filament, it helps the cell to make actin filament and more rapidly production of actin filament and much more stronger actin filaments. Okay. In other case, if it has the ARP complex, then it helps to nucleate an assembly to form a web and remains associated with the minus end. So what happens if it produces, if it attaches with the ARP complex, it's another protein complex, ARP. So this ARP protein complex will bind to this growing filament section, and after binding, it will help to make a web-like network of an actin filament. So it again is helping the actin filament to grow up, but to make a web-like structure. But in case of formin help in the actin filamentation via the production of straight chain filaments. But in case of ARP complex, it is making the web, a web-like structure. Okay. Now, if it has a thymosin, if it has a proline, first talk about proline. So if a proline protein associated with these actin subunits, then this binding of proline speeds up the elongation process of the production of this chain. So it will speed up the elongation. That means the much more actin along with profilin will come and bind with the positive end. It will elongate the chain of actin filament. But in another turn, if it bind with the thymosin, which is another one type of thymosin is called the thymosin beta 4. This thymosin will come in and bind with the subunit of actin filament, and it will prevent the assembly. So what we can see, we have the actin subunit, which are the monomer to make a polymer of actin filament. Now along with the attachment of different protein molecules, they have different effects. So if it attaches with formin or ARP complex, then they can use with each other to make a stack of actin filaments. In case of attachment to formin, it will make a stack of bundle-like actin assembly. In case of ARP, it will make a web-like assembly. In case of binding to profilin molecule, it will elongate really faster, and in case of binding with thymosin, the elongation will stop and it will go through the disassembly, or it will block the assembly of the molecule. It is in one turn. Now if we consider the matter with actin filament, now let's think about the actin filament and how actin filament will react with attaching with all these different types of molecules. If actin filament attaches with cofilin, so cofilin molecule come and attach with actin filament, then what happens? It binds the ADP actin filament and accelerates disassembly. So in this case, cofilin molecule will come and helps this actin filament to disassemble. So remember, all this profilin, cofilin, formin, thymosin beta 4 and all these things are working together to have a dynamic stability between this subunit form of actin and actin chain form. So there is this is a reaction, if you can see, from a monomer to polymer. For it's a polymerization reaction. In this polymerization reaction is having a different effector molecules, and these all proteins are acting like effector molecules. So cofilin will bind and helps in disassembly. In the same consequence, if you have gelsolin, it serves the filament and binds to the plus end, and it sever the filaments. Okay, and if it binds to the capping protein, that prevents the assembly and disassembly at the plus terminal. So capping protein binding will restrict its growth, but that means it will tell the cell that the length of the actin filament reaches the appropriate one. So if it needs to be the actin fiber for 10 suppose for example few nanometer, 100 nanometer suppose, so it reaches the 100 nanometer, then a cap protein will come in and attach to the positive side end. It will tell the cell that no longer you should stop now the growth of the actin filament, because it reaches the amount, it reaches the length it has to be reached. Okay, so this is the concept of capping protein. And also tropomyosin is an important protein that stabilizes the filament. So if tropomyosin come in and bind with this filament actin filament state, it will stabilizes the filament. So there are lots of different proteins are working, but remember one thing, they are all working to maintain the balance between the actin subunit as well as the actin filament construction. Okay, now here are different arrangement of these actin filaments. They can arrange like this fimbrine structure, they can arrange like this filamine structure, which is a web-like structure due to the help of ARP complex. They can attach with this pectin structure. This is another network of meshy actin filaments, and as well as they can form in this ERM structure. So there are lots of different structural variety that an actin filament can achieve, because an actin filament is abundant in nature. It can easily be achieved all these things. That's why now you can say why they use this actin filaments in such a different purposes, and why we can find this actin filament in abundant nature in the cell, because the actin can be modified into different things. So actin, if you have actin, you can use this to modulate different things, you can use this to maintain different things, and you can use this to do different works in a cell. So these are the different cross-linking and attachment of membranes. You can, in a membrane we can see. So it helps the cell membrane to get integrated, it helps the cell to maintain its structure, to regulate its structure. It also helps the cell to be motile, to get into a particular direction very clearly. So that's all the work of an actin filament. So actin filament is very important for a cell. Now let us talk about the intermediate filament and intermediate filament assembly. If we look at the structure of intermediate filament, it is made up with this protein molecule. It's an alpha helical monomer, we can see. Excuse me. So we have monomer with the N terminal and C terminal. So this N terminal C terminal monomer will attach with many of this monomer, at least here another monomer, to make a dimer. So we have a dimer having the same N and C terminal. So no distinguish. So it is a polar molecule now. So it is an N terminal C terminal. We call it a polar in nature. Now two of this dimer will attach with each other in such a way that they will eliminate this polarity. That means after the attaching of two dimers in opposite orientation, they finally make a construction of a polymer which is having both terminal with N. That means there is no polarity is shown in this case after the tetramer is formed, after the attachment of two dimers. So this tetramer arrangement is done. After the tetramer arrangement, this lot of different tetramers will come together and interact with each other by these head regions. These are called the head region, and this is the coiled coil region, which is the alpha helical protein structure. They pack together to make two tetramers will pack together to finally make a filament. Now this filament will attach with each other, they stack one by one with each other to finally make a filament structure like that. This is an electron micrograph showing the intermediate filament assembly. Now what is the work of an intermediate filaments? These intermediate filaments are really strong because they are made up with this alpha helical protein. They are fibrous in nature, they are really strong, so they help in integrity for a cell. So you have a cell lining, you have a different different number of cell align in a particular origin. So we have this basal lamina, and this lamina we have different you have many cells, and all these cells will be held together by this actin. So if we think about this, this is the intermediate filament. So intermediate filament go basolaterally, hold all these cells together. So that's a very important work for intermediate filament. Now we'll look into the animation of intermediate filament and how and why an intermediate filament is important. Now look.
Eukaryotic cells contain a complex network of filaments, intermediate filaments, microtubules, and actin filaments, that provide the cells with strength, structure, and movement. Although all eukaryotic cells contain microtubules and actin filaments, intermediate filaments are found only in vertebrates and a number of other soft-bodied animals. Intermediate filaments are found in animal cells that require a lot of strength, such as the epithelial cells of the skin. Some of these filaments span the length of the cell, connecting cell-cell junctions called desmosomes. These cables of intermediate filaments have a high tensile strength. Without these filaments, stretching or pressure on the epithelial sheet would cause it to rupture. Each filament is rope-like, consisting of 8 thinner strands made of a precise hierarchical arrangement of protein subunits. At the lowest level, 2 monomers associate with each other to create a twisted dimer. 2 dimers then line up to form a staggered tetramer. Note that the 2 dimers are arranged in opposite orientations, with their amino terminal ends away from each other, so that the 2 ends of the tetramer are indistinguishable. Tetramers then link end to end, thus building up one strand of an intermediate filament. A total of 8 strands stack together and twist around each other to create the intermediate filament. This stacking provides the extensive lateral contacts between the strands that give the filament its remarkable mechanical strength. An electron micrograph shows the appearance of intermediate filaments that have been assembled in a test tube.
Okay. So this was an animation about the intermediate filament. As we can see, the arrangement of the intermediate filament is typical in such a way that that can resist a typical huge tensile strength. Now we move on to the third kind of cytoskeleton. It is called microtubule. So yes, now let's talk about the microtubule. So what are microtubules? Microtubules are a third kind of cytoskeleton. They are made up with tubulin protein. So these tubulin proteins are, here is the structure of a tubulin protein. As you can see, it is made up with the 2 dimer. It is made up with 1 dimer, so 2 monomer, that means 1 dimer. It is a 1 beta tubulin, another one is alpha tubulin. So we can see the beta tubulin and alpha tubulin attach with each other. They have a binding site for ATP. But memorize one thing, that this beta tubulin is the most important monomer among this alpha and beta tubulin, because this beta tubulin can attach with ATP or ADP. So if beta tubulin is attached with ATP, that means if it is attached with ATP, that means it is active. And if it is attached with ADP, so I have some problem with my mouse, so it is very difficult to write down with mouse. Anyways, So if it is attached with ADP, that means it is inactive in nature. So what happens, this tubulin dimer will come in and attach with ATP. So ATP beta tubulin dimer is active, so this dimer will come in and attach with each other to finally arrange themselves in such a way. So one by one they arrange in a linear structure like that. Now one by one linear structure will come together, and if we make a sheet like this, then we fold this sheet up to finally give a three-dimensional array like structure like this. So we can finally end up with making a tubulin. So it's a tube. So tubulin is a kind of tube made up with tubulin dimer proteins, and this tube is made throughout the different throughout a sheet made up with this alpha and beta tubulin. The sheet is made by aligning one by one a strand of these tubulin proteins. Okay. So this is how the tubulin is arranged. We're going to see how this arrangement is done in the future slide with the animation. It will clear you concept very much. Now let us talk about the arrangement and how this arrangement is done. So if we have this dimer of alpha and beta tubulin, and this beta tubulin is attached with GTP. So I have talked about ATP previously, but this is not a ATP. It is very much important for GTP, because it is GTP bound protein. So if GTP bind to it, then it is active. In GDP bind to it, then it is inactive. So if GTP bind to the beta tubulin, it is active in nature. Now this active dimer come in, and this active dimer attached with another dimer, then it is attached with another dimer to make a polymer of tubulin. It is called the tubulin protein. A polymer of tubulin will be called a microtubule when the tubulin is arranged in a cylindrical arrangement. Okay, now here is the tubulin arrangement. Now after some time, when the GTP is hydrolyzed, GDP is made, and this tubulin will getting very much unstable, and it will be dissociated. Now why they are becoming unstable? Because of when the GTP bind to this tubulin proteins, then the tubulin is having a particular straight angle in their structural configuration, straight angle. But whenever GDP replaces the GTP, it's slightly tilted in some way, so it is slightly changed in its angle through its axis. That makes, you can see in this picture, it will bend their structure, linear structure in a few degree, and this bending helps to dissociate. So if we are looking at there, this is a growing phase of tubulin protein polymer, and this is a shrinking phase. In the growing phase, it is growing straight, but in the shrinking phase, what is happening when the GTP is hydrolyzed to GDP, then this microtubule B, tubulin B along with GDP is unstable, and they will enter into the state like this carved state. So they will dissociate like a peeling of banana like structure like that. So it can be easily seen in electron micrograph. I do not catch the picture of this electron micrograph, but still you can see in this dramatic representation that they can peel off like that. Okay, now what is at the assembly point? So if we think about the polymerization purposes, again in this case too, we can see the polymerization of lag phase, where we have we have simple tubulin proteins but no assembly of tubulin to make microtubule. Then we suddenly increases the growth rate and finally make them much more microtubules with those tubulin proteins, and after some time it reaches a constant, it reaches an equilibrium. In this equilibrium, no longer tubulin proteins will associate to make a microtubule, so it is stabilized at the state. Okay, it will reach the equilibrium phase. Okay, and what is going on in this case is either assembly or disassembly, and the assembly side is called again positive, and disassembly side is called the negative. In the assembly side, we have the GTP bind state of the tubulin, and then negative side we have GDP bound state of the tubulin. Okay, and in all this case, alpha tubulin plays a role of making the structure stable, but beta tubulin is making a structure of assembly and disassembly, or regulation of assembly and disassembly. That's why the beta tubulin is much more important. And these tubulin proteins are typical proteins which is helping the cell to divide. When a cell starts to divide, it needs to produce the array of microtubules. So this array of microtubules are called spindles, as you know. It calls spindles. The spindles will help to dissociate, to segregate the sister chromatids during the cell division. And the sister chromatid segregation is really important, because after the sister chromatid segregation, the new cell will be made, and in those new cell nucleuses, those one copy of the sister chromatid will go and to make a final copy of nucleus. That's why the sister chromatid division is really important. If anything wrong happens during the sister chromatic segregation, then it will end up with the abnormality in the chromosomal number. Okay, so that's why it's really important. Now let us look at the animation of microtubule assembly and disassembly. It is an animation narrated by Ivan Nogolus, and let's see.
Microtubules are dynamic cytoskeletal polymers essential for all cells. They are made of the protein tubulin, an alpha-beta heterodimer that self-assembles head-to-head, making protofilaments, which themselves associate laterally, making a cylindrical wall. Tubulin binds the small molecule GTP, or guanosine triphosphate. During microtubule polymerization, this molecule is hydrolyzed to GDP, or guanosine diphosphate. The GDP-bound form of tubulin is unstable in the microtubule and gives rise to the phenomenon of microtubule dynamic instability, by which microtubules switch between growing and shrinking phases. When microtubules lose a critical layer of GTP-containing tubulin subunits, shown in brown, they depolymerize by peeling and curving of the protofilaments. Although protofilament peels are short-lived, we stabilize them for structural analysis to define the tubulin arrangement and conformation within them. These disassembly intermediates break down into smaller pieces. We found that in the GDP depolymerized form, the tubulin dimer makes bent contacts but is also bent itself with a 12-degree angle. Upon GDP exchange for GTP, the tubulin subunit straightens, allowing it now to repolymerize, making both longitudinal and lateral interactions. The resulting assembly intermediate is an open sheet that is very short-lived, but we were able to stabilize into helical ribbons that were amenable to structural studies by electron microscopy and image analysis. This polymer shows the protofilaments arranged in pairs with contacts that alternate between those found in the cylindrical microtubule and new contacts never described before. The arrangement is said to be able to roll into a tube when it reaches the right width and length. The closing up of the microtubule is a highly cooperative zipping up process. Thus, microtubule growth occurs by sequential sheet extension followed by microtubule closure. Driven by the hydrolysis of GTP, microtubules switch between growing and shrinking phases via the structural intermediates just described, giving rise to a very dynamic cellular cytoskeleton that performs essential functions in the cell, from transport of cellular organelles to division of the genetic material between daughter cells during cell division. Microtubules are fascinating nanodevices that have evolved to undergo amazingly complex assembly processes.
Really nice animation. I hope every queries about microtubules is getting cleared after seeing this animation. And now we're going to discuss about the control of making this microtubule and the tubulin. So again we are talking about a polymer microtubule, which is a polymer of this tubulin protein. So tubulin dimers are there, and we are talking about a balance between making this tubulin and microtubule together at a particular time. And there again different effector proteins are there, which is helping to make to assemble and disassemble this tubulin proteins to make microtubule. And some of these proteins are gamma TARC is called is a typical protein which helps to nucleus and assembly of this tubulin proteins to make microtubule. Okay, so this gamma TARC most of the time seen in case of centromere. Centromere, remember, is a place through which is also called the microtubule organizing center, or MTOC, and through the centromere portion, this centromere uses this lots of this tubulin, and they arrange those tubulins together to make huge number of microtubules, because they need microtubule at the situation. Actually, these centromeres are the place which give rise to the formation of the spindles, which are being necessary for the cells to segregate the segregate the chromatids during the cell division. Okay. Now let's talk about stathmin. Stathmin is a kind of protein which binds to the subunit and prevents assembly. Stathmin binds to the subunit of this tubulin, probably the alpha subunit of the tubulin, and it will block the assembly of tubulin to make microtubule. Okay. Now let's talk about TIPs. It remain associated with the growing plus end and can link then either to the structures such as membranes, because sometimes the microtubules has to be attached with membranes because of the microtubule growth is needed in some cases, because the astral spindles which are being generated during the cell division from a centromere will be sometimes you have to interact with the cell membrane. In those situations, this kind of proteins like TIPs will help them to attach and link with the cell membrane, or different structures resemble to cell membrane. Okay. Now let's talk about some proteins which will interact with fully grown microtubule and how they modify their structure. So a simple protein is MAP, which stabilizes the tubule by binding to the sites. So MAP, what MAP does, MAP come and bind with this alpha and beta subunit of my tubulin proteins, and it stabilizes this microtubule structure. And we also have catenin protein, which severs microtubule. So this catenin protein is there to sever the microtubule, and we also have this XMAP215 is a designation of a protein which stabilizes the plus end of the molecule and accelerates the assembly. So the accelerate assembly is done by here the gamma TERK as well as XMAP. And MAP helps to stabilize the microtubule but do not helps to grow the plus end in very far. And now let's talk about the final kind of protein, which is called here is kinesin. Kinesin is a motor protein. So this motor protein enhances the catastrophic disassembly of the plus end. So it helps to disassembly of this molecule. So it helps in disassembly, and this helps in assembly. Gamma TERK helps in assembly, and XMAP also helps in assembly, and MAP helps in stabilization. So these are the actual work of different proteins along with microtubules. Some will interact with the tubulin dimer, some will interact with the fully grown microtubule. But the basic idea is to keep the equilibrium between the tubulin protein and microtubule in a particular constant manner. Okay. Now as we have talked about the kinesin, now let's talk about kinesin in much more detail. So what are these motor proteins? As we know, these motor proteins are really important during the cell division, because these motor proteins is actually generating the force of either assembly or disassembly during the cell division, where these microtubules are need to sometimes getting improved, sometimes getting dissociate or shrinked. So sometimes this microtubule has to be getting increased in length, sometimes they have to be getting decreased in length, which is called the shrinking. So this growing and shrinking, which we call the rapid disassembly nature, or we call the rapid dissociation nature of this microtubule, which is a characteristic of microtubule, which is helping the microtubule to grab and hold on to the hold on to those chromatids and segregate them, will need the help of this kinesin molecules. So what are the action of this kinesin molecule, how they work together? Now let's look at this picture. So what are these motor proteins? So if we see about these motor proteins, so whether the motor protein is kinesin, or the motor protein is dynein, or the motor protein we can call it will be myosin. Okay, so whatever motor protein we are talking about is working along with a different type of fibrous molecule. So if you talk about kinesin and dynein, they will act along with microtubule. So microtubule is a fibrous molecule, and kinesin and dynein will work on microtubule, help to move on microtubule. And if you talk about myosin, then myosin will help to work together with actin filament, with actin fibrous protein. Okay, so what they are doing simply, this picture illustrates what they are doing. So everything what is motile in the cell, we can see, will move from one place to another place. The cell components has to move. So if we have mitochondria, it has to move from one place to another place. If we have lysosome, it has to move. So every cellular vesicle trafficking which is happening all the time in a cell will have to move from one place to another place, is done by this motor proteins. So these motor proteins are the vehicles which actually work through the microtubule, sometimes through the microtubule network, sometimes through this actin network. So whether there's an actin network or microtubule network, we need different varieties of motor proteins to move on to them. So not only move on to them, but also to carry different things with them. So this to carry vesicle, for example, we have a vesicle, inside the vesicle we have the cargo protein. So you have the membrane glycoprotein and the cargo protein there. We need to carry this cargo protein from the for example from the Golgi bodies when it is sorted to the cell membrane to fuse this, because it has some cargo protein which is needed to be released outside the cell. So in this case it has to be transported, and this transport is being done, the carry is being done by these motor proteins. So in this case this carry is done by the dynein. So remember one thing, kinesin will move towards the positive end and dynein will move towards the negative end. So that this is the basic idea. Kinesin will move towards the positive end, dynein will move towards the negative end. And as kinesin is moved towards the positive end, it will pull the strand of microtubule in the negative end, and dynein will move towards the negative end, but it will pull the microtubule towards the positive end. So this is the basic concept. Do not forget this concept. Now let's talk about the myosin. Myosin is again a kind of motor protein, but it acts on the actin filament. It normally helps to generate the muscle strokes, as we can know. In muscle fibers, lots of myosin is there, which is always working on actin to generate the muscle stroke. Actually, this muscle stroke is helping us to work properly. Now let's talk about, focus on the structure of these molecules, the structure of these proteins, motor proteins. Normally what they have a chain of tail structure, then they have the head structure. So we have a head and a tail. Tail is a long, fibrous, and it's a rope-like structure. It's a coil structure of two helices, as you can see in this picture. And the head is made up with light chain and the N-terminal bulkier groups or bulkier regions like that. And these groups are called the bulkier head regions. These head regions actually have an attachment site with whether those fibrous proteins, whether it is microtubule or actin. So in this case, if you think about this, this is as a myosin, and then we have the actin fiber here. So this is an actin fiber, and this myosin head will bind. We have a binding site for this actin fiber, and as well as this will have another binding site for ATP, because all these proteins are ATP driven molecules, because all those motor which are going definitely needs energy to carry out the motor activation. So in this case also it needs energy, and the energy is being used via the hydrolyzing ATP. Okay, so it will hydrolyze ATP, so it has a binding site for ATP. This head has a binding site for ATP as well as a binding site for the actin filament. ATP will bind and it will be hydrolyzed to ADP, and the energy after the hydrolysis of ATP is used to walk through this actin filament. And how they work through this actin filament, how a kinesin will work to myosin filament, will be demonstrated by my next slide with a beautiful animation. Now let's go there. And this kind of movement is important, as I am telling you again, because without this movement the whole cell will become stagnant. Okay, now let's focus on here the motor protein movement animation. This is the animation of kinesin. So look at it.
The motor protein kinesin is a dimer with two identical motor heads. Each head consists of a catalytic core and a neck linker. In the cell, kinesins pull organelles along microtubule tracks. The organelle attaches to the other end of the long coiled coil that holds the two motor heads together. The organelle is not shown here. In solution, both kinesin heads contain tightly bound ADP and move randomly, driven by Brownian motion. When one of the two kinesin heads encounters a microtubule, it binds tightly. Microtubule binding causes ADP to be released from the attached head. ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the neck linker to zipper onto the catalytic core. This action throws the second head forward and brings it near the next binding site on the microtubule. The attached trailing head hydrolyzes the ATP and releases phosphate. As the neck linker unzippers from the trailing head, the leading head exchanges its nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats. In this way, kinesin dimers move processively, step by step, along the microtubule.
Okay, so that's an animation showing how motor protein will work through the fibrous protein and through a network of proteins inside the cell to keep on the motility of organelles inside the cell. Okay. Okay. Now let's talk about the cytoskeleton at work. That means in this final slide of my discussion we will talk about all those different proteins and how they work together to balance a cell, to balance the activity of a cell. So in this case we take an example of cell division. In this cell division we can see that all those kind of protein, microtubule, then actin filaments are maintaining the structure, intermediate filaments are holding the cells together, but in this particular cell division stage we can see the microtubules are assembling all the time, and those motor proteins are actually helping this assembly and disassembly, and as well as the segregation of all these chromatids together to be carried out in much more perfection. So you can see here the microtubules and the spindle pole is there. This is the chromatid, and this is the kinetochore region with which this microtubule spindle will attach and will destabilize and will take sister chromatids away. So here it is the dynamic instability of this microtubule, and sometimes after this growing and shrinking stage it will catch this kinetochore, and whenever the microtubule is attached to this kinetochore outer plate, then it will be attached, and it is giving a strain to this interaction between this junction of the chromatid, and the chromatin dimer disassembly will occur rapidly from this positive side end with the help of a protein, with the help of motor protein, because in this stage motor proteins will help to disassembly, and sometimes the motor protein will also help to assembly in this positive side. Okay, so we need the action of kinesin as well as dynein to work together. So we have a CNPE is a protein which will help in polymerization of the microtubule, and there is also dynein which will move towards negative end, so to help the shrink this microtubule. So this growing and shrinking will done using these motor proteins, and after the growing and shrinking phase it will be taken from both of the side. So if we'll take both of the side from here and there, so these strains will go on until they are arranged in a particular plate. We call this stage as a metaphase, when we arrange in metaphasic plate, and this arrangement, then the segregation will occur and all the things will work together. So that's how all these things are working together, all these proteins, actin filaments, intermediate filaments, as well as microtubules, and not only them but also the associate proteins, or what we call the motor proteins like myosin, dynein and kinesins are working together to maintain the cell work properly all the time. That's about the cytoskeleton of a cell, and I hope that's going to help you. Thank you.